Welcome to the genetics lab. The goal for this lab is to take a strain of Acinetobacter chalcoaceticus that is sensitive to the antibiotic streptomycin and see if we can change its genotype such that its phenotype makes it resistant to the antibiotic streptomycin. We're going to do this via a process called bacterial transformation. So in order to do that, I guess it's a good opportunity now to just quickly review what bacterial transformation is. In order for us to be able to do this, we're going to need to take our cells and get some DNA from that. And we're going to do that a very simple way using this thing here we call lysis buffer, which if you can see the bubbles, it's pretty much like soap water. Okay? So we're not going to measure out an exact amount. See, if you look at our microcentrifuge tube right there where it gets curvy, that's about half a milliliter. So just take a very cheap plastic pipette and then squeeze in approximately half a milliliter into your two tubes. There we go, something like that. And that'll be sufficient. As always, make sure everything is labeled. Your initials, and then S and R. This is one of those instances where, come on, focus. This is one of those instances where it's helpful to have one of those extra fine point Sharpies, right? Allows you to label better, but that's the one on the right over here. But clearly you can see on the left here, it works okay using a nice, not dull Sharpie, okay? These are going to be our DNA preps, and we're going to do it very simply. Take a loop. I was going to say tip, although you can use a tip. I do want to sterilize the loop because the loop is going to go and, oh, I guess you guys didn't see that. It's going to go and immerse itself into our buffer. I'm going to get this out of the way so we can focus. I'm going to take our streptomycin resistant cells and just take a healthy loopful. You don't have to take like this crazy amount, but you know, a healthy loopful. This is my streptomycin sensitive. Did I say resistant earlier? Here are my streptomycin sensitive DNA. And then I'm just going to work the cells off the loop and into my lysis buffer. Make sure we sterilize well. Just like that. Okay? And then, oops, I should sterilize. I'm going to repeat this for my streptomycin resistant cells. All right, that was a lot harder than it should have been. Now what we're going to do is we're going to do a very rough mix. So hold them like this and just shake them up. You'll notice that it'll get quite bubbly because of the soapiness, right? Ideally, we'd have a vortex here, but we don't have enough for everybody. So we're just going to shake it up. And then into the hot water bath. And when you are ready, you take your cell and lysis buffer suspension and bring it to the water bath. Simply put it in the rack, open the water bath, and place in. All right, so let's think about what's going on here, right? The lysis buffer, remember lysis means to break, has a little bit of soap in it, right? And what we're going to do is we're going to use the heat of the water bath along with that detergent, that soap, to break open the cells, right? It'll wear away at the membranes, cells will burst open. So moving forward, these are not cell suspensions anymore. These are what we're going to call our streptomycin sensitive DNA, streptomycin resistant DNA. But really what it is it's the DNA from the streptomycin sensitive strain of Acinetobacter chalcoaceticus and the DNA from the resistant strain, streptomycin resistant strain of Acinetobacter chalcoaceticus. We need a little bit of time for this to work, so we're going to close the lid and we're going to wait 30 minutes. About 30 minutes later, after 30 minutes, simply take out your DNA, not our cells. You can put this rack back, close it up, and head to your bench. While your tubes were in the Water bath, hopefully you spent your time wisely to label your plate as such. We have areas called resistant and sensitive DNA, resistant and sensitive cells, and then sensitive DNA, sensitive cells, resistant DNA, resistant cells on our TSA plate. All right, split it into 6, and then we're simply going to inoculate. Wherever it says resistant DNA, put our DNA we got from the water bath. Now I like to put the DNA first. Then I like to put my cells. And wherever you have an area where we mix our DNA and cells, I like to put the DNA first in that case as well. So here is my sensitive DNA. It should be kind of goopy. And then usually I just put like a small kind of circular amount in that spot right there. And then repeat for the other areas. And notice how I like to put my finger on the spot I'm about to spread, just so then I know which section to put it in, because of course when you lift your plate, sometimes you don't see the label and you can get lost. Okay, so now what I've done is I've placed the DNA onto our cells, onto our plate, wherever necessary. Now we're going to put the cells on top. So sensitive cells, resistant cells, and in the areas where we mix our cells and DNA, be sure to put your cells right on top of the DNA that you spread. Okay, so it looks like this. It's very important that the cells go right on top of that same patch of DNA. And that is the work for day 1. So the important thing today now is, before you leave, write down what are your expected results. What do you expect to see in each one of these sections when you look at your plates next time?